All right, everyone. It's time to continue and finish our last set of talks for the day. I'd like to introduce Holly O'Shea from University of Buffalo, who's going to be talking about developing a gene replacement therapy for STXBP1.
All right, good afternoon, everyone. I know it's late in the afternoon, so I appreciate you sticking with me. I hope you're kind of awake still. But my name is Holly. I'm a postdoctoral associate from the University at Buffalo, and I'm here today on behalf of my PIs, Sue and Jay, who work at the FOXG1 Research Center, to talk about some of our recent work into gene therapy development for STXBP1. And we are very new members of the STXBP1 community, so we're really excited to be here today. And a big thank you to the foundation for having us. So just to give you, since we're so new, a little introduction into who we are as a group and kind of how we got here. So my PIs are Sue and Jay, pictured here with their beautiful family. And Sue and Jay have been working in the field of neurodevelopmental research for a very long time, but their focus shifted when 16 years ago their daughter Yuna was born, and they found out that she had a disease called FOXG1 syndrome. And that is similar to STXBP1 in the sense that it's also a monogenic neurodevelopmental disorder. It has a similar incidence rate to STXBP1, so it's quite rare, and it causes some pretty severe clinical symptoms. So Sue and Jay were kind of left in the situation of a lot of our STXBP1 and all of our rare disease families where your child has been diagnosed with this disease, we don't know a lot about it, and there are also no good treatments for it beyond symptom management. So this led them to shifting their research entirely to focusing just on FOXG1 syndrome, and they last year established the FOXG1 Research Center at the University at Buffalo. And through our work, I've been with them for the past 6 years, but they've been working for a bit longer than that. We've been able to collaborate with also our parent-led organization, the FOXG1 Research Foundation, and develop a gene therapy vector for FOXG1 syndrome, which we were very lucky to get approved by the FDA this year. So clinical trials will be starting for that later this year. We're kind of anxiously awaiting all the clinical sites to get approved. But we were just very excited by this experience and being able to kind of be a parent-led organization and kind of work with other parents to get a treatment to the clinic, because we know how hard that can be. So we were very excited now at this point to kind of share our experience with other parent-led rare neurodevelopmental disease organizations to try to help other parents and other groups get these treatments to the clinic. And that's what led us to connecting with the STXBP1 Foundation this year. So we're very new to STXBP1 research, but again, very grateful to be here. So one thing we want to think about when thinking of therapeutic development for any disorders, but in this case STXBP1, is what is the primary disease mechanism that we can really attack here. And as we've talked about quite a bit today, this is predominantly thought to be haploinsufficiency in the case of STXBP1. So we get reduced gene dosage, which leads to insufficient STXBP1 protein production to sustain our healthy neuronal function. And as we've discussed, though there still could be further work in this area, there is no current evidence of patient variants inducing pathological gain of function or dominant negative effects, making STXBP1-related disorders a really good candidate for gene replacement therapy. So our goal when designing gene therapeutics, kind of broadly, is that we like to consider that we don't necessarily just want to drive very high STXBP1 expression. We also want to be careful to supply the proper dosage of the gene to the cell populations that really need it. So this requires a delicate balancing act. If our expression is too high or too broad, meaning if we target tissues in other places where we don't want it, we may trigger some sort of stoichiometric imbalance or cellular toxicity. And if our expression is too low or too narrow, we may not reach a sufficient fraction of neurons to drive phenotypic rescue and meaningful symptom relief. So with that in mind, we've been working with the foundation to design a total of 4 viral cassettes that range in anticipated specificity, and we're now working on testing them head-to-head to kind of see what is this balance between expression level and also region. So with that in mind, we kind of came up with 4 distinct combinations. So the first one, these kind of go from broad to more specific, is we designed a cassette with a ubiquitous promoter that's expected to drive STXBP1 expression everywhere. And then we restricted this a little bit with our second construct where we still have that same promoter, but we added a neuronal restriction element. And then finally, we have 2 cassettes that include distinct combinations of endogenous STXBP1 promoter enhancer elements, trying to kind of more so recapitulate the native conditions of STXBP1 expression. So our goal here with these studies is to work on mapping the trade-off between overall expression level and also cell type specificity. So for these studies, we chose to kind of do an initial screen of what kind of expression we get with these viral cassettes. So our route of administration here was ICV injection, and in this case, we also did bilateral injection to try to make things symmetrical, and we used wild-type mice, specifically C57 pups, at about P0 to P1. So we injected these wild-type pups with each of the 4 viruses at an equivalent dosage, and then just let them hang out for a month. And then 1 month later, we harvested their brains at P30, and then we have sectioned those samples and performed, kind of as our starting point, multiplexed in situ hybridization analysis, specifically via RNA-scope. And the benefit of the way we have designed this experiment is that we're able to simultaneously assess both endogenous mouse STXBP1 and also our vector-driven human STXBP1 expression without cross-reactivity. So we can look at our endogenous STXBP1 levels and then how is our viral-driven STXBP1 responding to that. So these are what the results looked like for our first construct. So again, this is the ubiquitous promoter. This is a coronal section of a mouse brain with a bunch of kind of key forebrain areas labeled here. And we can see that endogenous STXBP1 shown here in green, so our mouse STXBP1 is expressed pretty robustly throughout the entire brain in a lot of these key areas like the cortex, striatum, hippocampus, thalamus, and hypothalamus. And then also our exogenous STXBP1, so our viral-driven STXBP1 was expressed pretty robustly. So we saw good STXBP1 expression from our virus, and this was also quite broad throughout the brain. Our next construct was that same ubiquitous promoter with the neuronal restriction element, so in this case, looking specifically at transducing neurons. And again, we saw really nice transduction with our viral vector, so we get good expression of STXBP1 throughout the brain. And we noticed that with these samples, it looked even better and more robust than our ubiquitous promoter alone. And then getting into our more specific combinations, this was our first endogenous promoter enhancer combination. So again, we saw pretty good, robust expression of exogenous STXBP1 here in areas that are comparable to our endogenous mouse STXBP1 expression. And then finally, our most restrictive construct is kind of where we saw the biggest difference. So this is another endogenous promoter enhancer combination. And in this case, we still got viral human STXBP1 expression, but it was a lot more regionally restricted. So in the cortex, it looks a lot more deep layer specific. And we noticed our highest expression in the thalamus and some of our deeper brain regions. So just to give you a sense of how these compare to each other, this is just all of the viral expression next to each other. So our more broad constructs, kind of as we anticipated, drove more broad expression of human STXBP1 versus our more specific constructs were a bit more reduced spatially. And this is just how that compares to the endogenous mouse STXBP1 expression. So zooming in a little closer just to show you some of these brain areas. So we have kind of on the top here a bit of the cortex and then a bit of the hippocampus towards the bottom. So we generally saw, as we expected with our ubiquitous promoter combinations, we get good overlap of endogenous mouse STXBP1 expression and our viral-driven human STXBP1 expression, but there are also some cells mixed in here which are exclusively red. You can kind of particularly see those in the hippocampus. And those are expressing our viral-driven human STXBP1, but not endogenous mouse STXBP1. And the same was true for our ubiquitous promoter with the neuronal restriction element. And then with our endogenous promoter enhancer combinations, we saw a lot more specific overlap between the endogenous mouse STXBP1 and our viral-driven human STXBP1 as well. So a lot less to pretty much none of those cells that are exclusively red. This is also, I think, even more clearly visible in some of our deeper brain areas. So we kind of have a look at the thalamus on the top right and the striatum on the bottom left. So with our ubiquitous promoter, we're getting these cells that express just the viral human STXBP1, but not endogenous mouse STXBP1. And with our endogenous promoter enhancer combinations, even though the expression is more restricted, we see a lot cleaner overlap. So in total, all of our viral constructs successfully drove human STXBP1 expression as detected by in situ hybridization. Constructs 1 through 3 drove the strongest mRNA expression, and construct 4 also drove mRNA expression, but it was more regionally restricted. But despite this regional restriction, the increased specificity in vector design did lead to a higher overlap between our vector-driven human STXBP1 and endogenous mouse STXBP1 expression. One surprise to us was that based on some of our prior work, we had expected the ubiquitous promoter alone to drive noticeably higher expression than all other constructs. However, it wasn't necessarily the standout. If anything, the ubiquitous promoter with the neuronal restriction element drove a bit better expression. So just kind of our lesson here is something that we know, but it just really reaffirmed to us that you can't predict viral efficiency from promoter strength on paper. These in vivo experiments are really necessary to truly assess performance. And another important lesson to consider that we've talked quite a bit about today is that our mRNA levels that we're seeing here are not necessarily going to be equivalent to protein production. We may have inherent regulatory mechanisms or stoichiometric constraints that are capping our protein production even if abundant mRNA is produced. And this may be particularly true when assessing viral performance in a wild-type background like we are here. This is not really mimicking our patient condition. So ideally, we would like to do a definitive protein readout in a mutant background. So we're quite early on in our design and kind of testing process, but looking at our immediate next steps and kind of the broader path that would take this to the clinic, is that we want to quantify protein production via both Western blotting and immunohistochemistry in these samples. Again, see what is the actual protein induction that we get compared to mRNA. We've gotten a couple antibodies that we would like to test out, but if anybody in the room has recommendations, I would love to hear it. And then we also want to more specifically map our cell type specificity with established markers. So we're seeing here kind of generally, spatially, we get good viral induction, and we can kind of generally map that to the brain areas, but we haven't looked specifically at what these cells are that are expressing the exogenous STXBP1, particularly those ones that are not expressing the endogenous mouse STXBP1. So we would like to look more carefully at cell type markers and kind of see where we're getting that expression. We also would like to look at overall viral efficacy and tolerability. This would be a later step, and ideally this would be done in an established STXBP1 mutant model. But we may see that our vectors successfully drive STXBP1 expression. If we see that with mRNA and protein, that's fantastic, but do they actually promote meaningful phenotypic rescue? And do we observe any undesired side effects based on their expression? And from here, our kind of very long-term goal would be to narrow down our 4 candidates to 1 to 2 potential leads and proceed with further clinical preparation and investigation, which could include things like dose-response finding studies, more in-depth toxicology and biodistribution studies, and things like that. So with that, I would like to thank, of course, the Foundation for supporting all of this work that we've been doing, as well as my PIs, Sue and Jay, and all of our lab members, particularly those who contributed to this work, including our former postdoc, Chian, and 2 of our current PhD candidates, Varsha and Dongjun. And with that, I'm happy to take any questions.
Ollie, thanks so much. Really great talk. You were talking about the endogenous promoter. Can you enlighten us, or is it?
I don't think I'm at liberty to give too many details.
Yeah,
Sorry about that.
I had another question. So your RNA scope is really great. What is the resolution like? Can you zoom into synapses? We were talking about RNA, STXBP1 RNA being present at synapses potentially. Is that something that you can assess, or is it?
I think it is possible generally with RNAscope. We haven't specifically tried that in brain sections. It can be a little harder to visualize just because of how crowded all of the cells are, but definitely something we would like to look into. We've only done this kind of lower mag imaging so far, so we could we have a confocal, we can get up to at least like 60x and kind of look at that more closely. I think that would be really nice to see.
Would be great. And we have antibodies for you if you.
Thank you.
What was the dose you injected into the animals?
We did 5e10 viral genomes per microliter, and that was the same for all of the constructs that we tested.
And how much did you give to each mouse? 1 microliter to the mouse, or?
That was the total dosage for the mouse, so a total of 5 microliters, 2.5 into each hemisphere, because we did bilateral injection. Yeah. Thank you.
Yeah, nice work.
Thank you.
I was just wondering, are you also planning on potentially doing ICV or ICM injection in humans, or would you, I mean, I don't think, did you show the capsid?
We did not show the capsid, so in this case, it's PHP-EB at the request of the foundation, so that's going to be murine specific. We would need to decide a clinical capsid to move forward into human testing. It would be, I mean, that would be a further discussion, I think, with the foundation in terms of what our exact route of administration would be in human patients. In this case, we did ICV in mouse. We have the capabilities to test ICM as well, and at least with our prior work, we are going to be administering it via ICV in human patients. So we'll, I guess, be able to see how that goes as a bit of a preliminary study, and then kind of discuss further what the best route would be. But I think there are a lot of advantages to ICV, but also there can be a lot of concerns as well with that being a much more invasive procedure. So something to be considered for sure. Thank you. Thank you.